everyone. Thank you for joining today. So I would like to talk about the machine and the ghost. So here we're talking about the ghost is the default mode network, that really mysterious functional network that gets a lot of press in functional imaging. And we're trying to understand what powers this based on cytoarchitecture. Let my slides have a second to start running. Oh, here we go. Okay. So I think it's been quite interesting to note how our conceptualization of the default mode has changed over the past 20 years. It was first described as a task negative set of regions representing this offline mode of the brain, but this was subsequently challenged by evidence of enhanced activity of the default mode during tasks that draw on memory. Things like context-specific decision-making or narrative comprehension or social cognition. And all of these tasks, they share this dependence on integration of information from internal as well as external sources. So there's several contemporary theories have started to postulate that internal and external information is balanced by the default mode. And this is really a core aspect of its function. So you can see this, for example, from a recent review by Yuri Hasson's group. In this schematic, the default mode is positioned so it integrates incoming extrinsic information, it integrates that with prior information, and then it is thought to enable the formation of rich context-dependent models of a situation as they unfold in time. Now that's based on task fMRI literature. In the resting state literature, we tend to find the default mode conceptualized as the apex of a processing hierarchy. For example, in stepwise functional connectivity, if you seed from a primary sensory area and let the algorithm progress, it ends up finding the default mode, and there it achieves a new stable state. Also, if you look at the principal axis of differentiation in resting state functional connectivity, see on one side it's anchored by sensory regions and on the other side by the default mode. So both these methods emphasize the default mode as an extreme opposition to primary sensory areas. And also more so topographically, we see that the peaks of the default mode here are maximally different from primary sensory areas. So in a geometric sense as well, the default mode is seen as an extreme opposition to primary sensory areas. So we set out to test these hypotheses and provide a richer characterization of the default mode from a neuroanatomical perspective, which we found was really lacking in the literature at the moment. And we have this perspective of neuroanatomy as well as ideas of parallel distributed processing. So we wanted to ask, what is the default mode made of? What's the cytoarchitectural machinery powering this ghost? What is the position of the default mode in the brain? How is it connected? And then this can hopefully give us a better idea of what information enters the default mode. So we started by looking at the composition of the default mode with respect to cortical type. So these were originally defined by von Economo, and they were recently reanalyzed by García-Cabezas et al. And we were particularly interested in these cortical types you see here because they index the sensory fugal hierarchy that was described by Meslem. So here you have cortical organization described as a series of rings. And as you move inwards, you have decreasing laminar differentiation. So you have conical cortex on the outside. So you have a very prominent granular layer and many sublayers as well. And then as you move inwards, the prevalence of that granular layer decreases. So you have decreasing granularity as you move in. And theoretically, this is synonymous with changes in the function as well, from primary sensory to unimodal to heterolimbic to paralimbic and limbic. So this can provide a cytoarchitectural proxy for proximity to the external world versus the internal milieu. We found that the default mode harbors all 6 cortical types, signifying marked cytoarchitectonic heterogeneity. And then chi-squared tests showed that the cortical types were not equally represented within the default mode, however. So we compared the proportions of cortical types in the default mode to 1,000 randomly rotated versions of the atlas. This preserves the size and the shape of the default mode. And we found that the default mode overrepresents eulaminate 1. This one here, kind of a heteromodal type. And it also underrepresents conical cortex. We could guess that the conical cortex is only included in default mode because we're using atlases rather than subject-specific maps of cytoarchitecture and function as well. But overall, we see that the default mode is really cytoarchitectonically heterogeneous. But this variability is more nuanced than just types. So that's where we brought in BigBrain. So we mapped the cytoarchitectural variability within the default mode by measuring intracortical variations in cell body staining from BigBrain. We had a total of 62,960 staining intensity profiles. These are little vertices all over the brain. And so these are in the direction of cortical columns, and they see depth-wise variations in cell density and soma size. Then we looked at the principal axis of variation in cytoarchitecture using diffusion map embedding, which is a nonlinear manifold learning technique. And this first eigenvector explained approximately 42% of variance, and it illustrates this really marbled pattern of cytoarchitectural differentiation. So this first eigenvector, E1, I'll call it, it captures changes in the intracortical staining intensity profiles. So these blue areas you see all over the brain, they have a higher overall staining intensity and also this marked bump towards the middle as well. Whereas as you move to more positive values in the red colors over here more, you see that the profile is more flat and there's less differentiation across different depths. So then each subregion of the default mode comprises a broad range of E1. So you see this based on this, the rain cloud plots, that each subregion, so parahippocampus, inferior parietal lobule, middle temporal gyrus, inferior frontal gyrus, precuneus, and prefrontal cortex. So they all have the full range of this eigenvector within them. So they all have this cytoarchitectural diversity within them. But we started to notice just visually that they express this variation in very different ways. So you see here in the parahippocampal gyrus, there seems to be a more smooth gradient moving across E1. Whereas if you look in this prefrontal cortex area, there's periodic changes from extremes of E1. So this looks more like a pattern of interdigitation that we know exists in the prefrontal cortex as well. We also see these patterns of interdigitation in inferior frontal gyrus and to some extent as well in inferior parietal lobule and in middle temporal gyrus. Whereas there's a slightly different pattern once again if we look at cingulate linked areas. So if you look at anterior cingulate and middle prefrontal cortex or precuneus type area, you tend to see a gradient as you move outwards from the limbus, but there's striations within this. You can see those if you look closely in here as well. And we found that these presence of these striations was not related to curvature necessarily. So it seemed to be something really related to the cytoarchitecture as you move away from the limbus. So next we wanted to quantify these differences in the spatial patterns, and we stumbled upon this very cool niche of literature from landscape ecology, and that then led us to the concept of roughness parameterization from material physics. So these are a set of parameters. They're relatively simple calculations, but they tell you about differences in the amplitude and the patterning of a textured surface. So here we use E1 as the texture or elevation of the cortical landscape. So here you can see the parahippocampal gyrus as an example, where we flatten it essentially. So we use a surface mesh, and then we use this eigenvector to say the height of this landscape. And then we can look at the roughness of this type of landscape. So we wanted to see which type of parameter best differentiates the different subregions. And we found that the parameter with the highest coefficient of variation was the number of intersections through the zero plane. So the number of times it jumps from a positive to a negative. So as you can see in parahippocampal gyrus, it does that very few times because it's just this linear gradient necessarily. Whereas in more prefrontal areas where you see very high number of intersections, that's because it's jumping back and forth between the extremes of E1. So, and then overall, we actually found greater variation across the spatial parameters of roughness rather than amplitude parameters. So this complements what we saw qualitatively, that subregions differ in the spatial patterning of cytoarchitectural differentiation more so than their cytoarchitectural content. Then using Atlas overlap approach, we also find that all the networks encompass multiple cortical types, albeit with varied distributions. As you can see here, the ventral attention was most similar to the default mode in the makeup. And also when we use BigBrain here to look at cytoarchitectural similarity within each network, we find that the default mode has the highest heterogeneity, but it's closely followed by ventral attention and the limbic network. So now we want to build up this neuroanatomical characterization of the default mode in terms of its intrinsic architecture, but also regional differences in extrinsic connectivity. We already know that the default mode occupies this unique position in a cortical mantle. So we want to ask how this topography influences of regional differences in connectivity of the default mode to the rest of the brain. And to do so, we combine diffusion-based tractography with distance to calculate navigation efficiency between 400 parcels across the whole isocortex. So navigation efficiency really elegantly fuses distance and tractography together. And there's also research showing that it closely approximates invasive track tracing connectomes in non-human primates. So I think it's really well suited to map low-cost wiring in the brain. Then we can return to this question of whether default mode balances communication from external and internal sources. So here we looked at navigation efficiency of the default mode to different cortical types, using these cortical types once again as a proxy for relationship to either the external world or the internal milieu. So here I'm showing median navigation efficiency of each parcel to each type, and we've stratified the parcels by network. So each bar captures the range of navigation efficiency from parcels in that network to a specific type. So in some networks, there's obvious favoritism. In the limbic network, you can see that there's very strong navigation efficiency to dysgranular type. But in the default mode, you actually see this balance. And compared to null models, the default mode was the only network that seemed to have a real balance in navigation efficiency to all the different cortical types. So it shows that default mode has this potential to equally communicate across the sensory-fugal hierarchy. Then we want to test whether cytoarchitectural axis contributes to this balance, and average navigation efficiency decreases along E1. I should point out here that we use BigBrain Warp to transform from this cytoarchitectural axis that we defined on BigBrain to FS average as well, because here everything is in vivo imaging in this section of the talk. And I did a talk a couple of days ago on BigBrain Warp if anyone's interested in more about that toolbox. But back to the results. So we have that there's a decrease in navigation efficiency across this cytoarchitectural axis. And this effect is especially pronounced for more granular types, as you see here. So the lower E1 values have especially heightened navigation efficiency to more externally focused areas. So in essence, we find that connectivity of the default mode is organized along that cytoarchitectural axis. Now we've learned that this structural manifold of the default mode has a potential for equipoise communication across the sensory-fugal hierarchy. But understanding how this potential is actualized requires really examination of ongoing fluctuations in brain activity. And to do so, we modeled the effective connectivity using resting state fMRI and regression dynamic causal modeling. This is a really scalable generative model of effective connectivity that can be performed on the whole brain and lets us inspect directed signal flow. So first we looked at this type effect again, and we find that the balance is present in the efferent connectivity, so going out of the default mode, but not in the afferent connectivity. So DMN broadcasts widely across the sensory-fugal hierarchy. Then we look at the axis, and we find that average afferent connectivity decreases, but there's no such effect for the efferent connectivity. So negative correlation, the afferent connectivity mirrors what we saw in the navigation efficiency. So once again, that the high E1 is more insulated from incoming information. But unlike the navigation efficiency, we do not observe a type by axis interaction. So when we combine this fMRI-derived afferent connectivity with the navigation efficiency results, we could hypothesize that the low E1 aspect is this aspect here. It's receiving the majority of the input from the default mode, and based on the navigation efficiency, it seems that the more granular types could have faster communication or higher fidelity information. Okay, so our description of the default mode's neuronal architecture hopefully will help us to begin formulating a theory on what information enters the default mode and what the default mode does with that information. So input to the default mode is organized along the cytoarchitectural axis in such a way that there is preferential input to the low E1 aspect of the axis. So this can act as convergence points for information from across the centrifugal hierarchy. And these convergence points could allow for a unique reconstruction of information. And this seems to kind of light a fire within the default mode. And it leaves this one section of the default mode relatively insulated from input. Then once we're into the default mode, we see that there's really vast cytoarchitectural heterogeneity, and we know from a lot of previous research there's really dense interconnectivity as well. So we see here that the information is constantly transformed within the default mode. It's changing its dimensionality, it's reconstructed as it moves through these different types of cytoarchitectural forms. And the default mode seems to reconstruct information in a distinctive manner that's not enabled by classic centrifugal hierarchical processing streams. And finally, we find that the default mode then widely broadcasts its information back across the centrifugal hierarchy, which speaks to its potential to shape our prediction at many levels. So with that, I'd just like to thank everyone who's been involved with this process, everyone who's supported me along the way, especially my mentor, Boris Bernhardt, and Margie Garber, who was a wonderful PhD student in Boris's lab who helped start this project off. And I'd be happy to answer any questions.
Casey, thank you for such a lovely and marvelous talk. Let me see if I can get myself here. There we go. Okay. I'm going to interpose with a quick question and then give to the audience. I have often thought from macaque connectivity that there's a higher degree of branching in what I would call the association areas, but I'm quite willing to think that it actually could be from the default network as well. So if I take you to the question of individual axons branching among the areas, would you care to comment on the possibility of higher degree of axon branching?
Yes, definitely. What we've seemed to find in the default mode is it may comprise areas of high axonal branching, but then that is complemented by areas possibly with lower axonal branching, especially that we're seeing, for example, in the prefrontal cortex, this interdigitation of different types of cytoarchitectural forms. So I think that would be consistent with some areas with very high axonal branching, neighbored to areas with lower axonal branching.
Yes, it could be.
Yeah, I think that would definitely be consistent with what we found.
Okay, love to talk with you.
Yeah.
Okay, but audience, questions, please. I think we have some time. Okay, well, then at the poster session, or let me see, poster session or later on, Casey, thank you ever so much. That was lovely.
I have a question.
Oh, yes, Alan.
Wonderful talk, Casey. I was wondering if you've seen any evidence in your studies here of a rich club of hubs.
What we do see is that there's some areas of the default mode that are receiving a lot more input than others. So it doesn't seem that the entire default mode is a rich club, but there are subcomponents of the default mode that are a rich club. Because there's some parts of default mode that are quite insulated from input. Those areas also have greater output to the basal ganglia. So it seems there are some parts of default mode that receive a lot, a relatively high level of input though.
And there is a question from Nicola.
Hi, Casey, lovely talk. Let me bring my hand down before I forget it. Great talk, really nice talk. I've got a question concerning that patchy appearance that you described in the prefrontal cortex, and especially in the lateral prefrontal cortex. And I was wondering, and as opposed to this gradual changes that you described, for example, in the posterior cingulate cortex. So this is a provocative question, right? Could this have to do with the methodical problems that we have in sampling cortical layers and the fact that, you know, we know that cortical thickness changes with the folding and so on. We do have all of these little sulci and so on in the lateral prefrontal cortex, which aren't present in the posterior cingulate.
I was also very concerned about that.
Right.
Because and so what we did try to do was look at the curvature of the prefrontal cortex and see if that matched on to this pattern, and it doesn't seem to actually.
Right.
So
well, that's
now,
reassuring. So
yeah,
we don't understand it, but it would exclude the worry. Okay.
Yeah.
Nice. Well then, yeah, that makes.
I also thought
that's a fascinating
Yeah.
finding, and it makes it more difficult to understand.
Yeah, possibly.
I think Sebastian also found like islands in the frontal lobe, which could be. I mean, and with islands, I don't mean the cell clumps like that we find in layer 2 of the entorhinal cortex. I mean,
probably something like cytochrome oxidase stripes.
Yes, exactly.
Yeah,
exactly.
I could. Okay, this is marvelous.
Kathleen, hi, it's Boris speaking here. So
Yes.
there's just a recent preprint, so just following up on your question from the Van den Heuvel group, and they showed branching complexity of dendrites based on postmortem sample and associated them with complexity of diffusion MRI tractograms. So it's different data sets, obviously, but there is this evidence that the branching complexity increases along the hierarchy.
Oh, yes, I think there's a lot to say.